Hello everyone, my name is Simon from the Vallée-Belisle Group at University of Montreal. Today we'll be going over the DNA synthesis procedure we use in our lab.
This experience enables a DNA sequence synthesis. Oligonucleotide synthesis is the chemical production of relatively short fragments of nucleic acid with defined chemical structure sequence. Typically, synthetic oligonucleotides are single-stranded DNA up to 100 mers in length. We first enter the sequence we want to create. The sequence is written in a 3' to 5' direction. We use purified phosphoramidites and columns for the synthesis. The first step is to add the column and start the sequence. The phosphoramidites are in bottle solutions, and columns have a DMT group and links the first nucleoside base. The chemistry cycle relative to the synthesis is 1. Activation and coupling, 2. Oxidation, 3. Deprotection, and the final step in the cycle is the capping step. The machine will pump different solvent to realize the cycle. We can quantify our experience with the yield and V2 value. After the synthesis, we need to cleave our sequence from the column by putting the column in ammonium hydroxide overnight at 60 degrees. We then need to purify the DNA sequence to only extract our wanted sequence. This is done by RPC, reverse phase chromatography. Solvent with different hydrophobicity affinity will help us target and elute our sequence from the sample. After RPC, we have to dry the sequence in a Speedback at 55 degrees Celsius for 3 hours to evaporate the acetone nitrile. The last step is DNA quantification. This is done with UV-Vis using a nanodrop machine. It allows to measure small sample volumes. Molecules have different emission intensity at different wavelengths, like salt, DNA, and proteins. Using a ratio of absorbance at 260 and 280 nanometers, we can quantify the concentration and purity of our DNA sequence.